Thank you. Can you hear me and see my presentation okay?
Yes, we can see you and we can hear you and see your presentation. And it's not in presentation mode currently.
How about that?
Not perfect. Thank you.
Okay. Well, first of all, thank you for the kind introduction and thank you to the CBA for inviting me to speak. I will get right into it. Um, Chin gave a really nice introduction that really covers a lot of the introduction that I needed to present. So I will be very brief in the introduction. But ADCs are a very compelling drug modality for the treatment of cancer that allow for the targeted delivery of really potent cytotoxins to cancer cells. And their role is to specifically deliver to cancer cells on one hand, but also protect the body, the normal cells, from the cytotoxins that they carry. And so by combining these two functions, we can get an enhanced therapeutic index from our ADCs. And while the concept is really interesting and makes sense rationally, it's been extremely difficult to generate ADCs that work well in the clinic. In fact, Dr. Lee has already shown this slide. There are a number of different ADCs that are currently being studied in clinical trials, over 240 at the moment, but only 17 have been FDA approved and are currently available in the market. Most of the ADCs failed during clinical trials because of a lack of therapeutic index. And so it remains a significant challenge to try to solve this problem. So of the 17 antibodies that are approved by the FDA, all of them are constructed using what I call conventional conjugation chemistries. So these are chemistries that target either the cysteines, the reduced cysteines of antibodies, or the amine groups of lysines. Both of these chemistries are very robust and very efficient. However, they do generate antibodies that are heterogeneous, and by attaching hydrophobic payloads to them at exposed sites, we tend to generate very hydrophobic ADCs. So the lysines, for example, an antibody carries about 40 lysines per heavy chain. So there's 80 potential sites of conjugation. And by stoichiometrically controlling the payload numbers, we can control somewhat the number of payloads that we attach to an antibody, but it's heterogeneous and we generate a lot of regioisomers. Cysteines are a little bit better. There's only 8 accessible interchain cysteines to conjugate to. So a DAR8 tends to be more homogeneous, but the payloads are exposed. And if they're hydrophobic, they tend to drive the hydrophobicity of the ADC. By titrating down the amount of payload, we can generate ADCs with lower DARs between 2, 4, 6. But the cysteine chemistry has one flaw in that chemistry, the thiolmalamide chemistry, is reversible. So we tend to see some loss of payload in vivo. And so these limitations have a functional impact on ADCs. And that's one example is shown on the right, where, and this is the pharmacokinetic curves of an antibody, a naked antibody at the top with green. As soon as we attach hydrophobic payloads to the surface of an antibody, we increase the hydrophobicity and we see accelerated clearance of that antibody. If you consider then that the payloads might fall off the antibody as they circulate, we see even further decrease in the available antibody or ADC that is available to reach the tumors. And so by decreasing the effective numbers of ADCs in circulation, we also impact their overall efficacy. So making antibodies, ADCs, isn't really difficult. Conjugating them, there are many different methods for doing this. We've talked about amine coupling and cysteine thiol conjugations. There's a number of different enzymatic approaches. You just heard Dr. Wang talk about glycoengineering and glycoremodeling to enable bioconjugations. And then there's also the use of non-natural amino acids to generate, all of these help to generate site-specific ADCs that have higher homogeneity and overall improvements in their biophysical properties. And one good example of how these improve homogeneity and improve stability of the ADC impacts an ADC is shown on the right from this paper from Pfizer from 2020, where they made lysine-based conjugates that are more stable and more homogeneous, and we see improved PK and improvements in their overall safety. Okay, so my team is interested in understanding how the sites of conjugation impact the function of an ADC and the overall properties and in vivo performance of an ADC. So we chose to use non-natural amino acids to generate our ADCs. And the reason is that that gave us the most flexibility for protein engineering. And to incorporate a non-natural amino acid, it requires a little bit of cell line engineering. So to briefly explain, during translation, a ribosome will run along an mRNA, and as it reads the codons, it recruits a tRNA that has an amino acid attached to it. And the ribosome will connect the arriving tRNA, amino acylated tRNA, to the growing polypeptide chain to form a full protein. Most cells have 20, one of the key controlling enzymes of protein translation is a set of enzymes called amino acyl tRNA synthetases. It's shown here like this bean in blue. And their role is to recognize a specific amino acid and a specific tRNA and to connect the two together. And cells have 20 of these amino acyl tRNA synthetases, one for each amino acid. So in order to encode a non-natural amino acid, what we've done is we've engineered cells that have a 21st amino acyl tRNA synthetase with specificity for a non-natural amino acid and its cognate tRNA. And that tRNA is directed to amber codons that we can encode at any position on an mRNA and control precisely where that non-natural amino acid is placed. So why a non-natural amino acid? And what is a non-natural amino acid? A non-natural amino acid is an amino acid that has a functional group that is not found in nature. And in our case, we like to use this amino acid called the lysine cyclopentadiene. So it's lysine up to this amine group here. Then we have a carbamate and this cyclopentadiene that enables a really nice reaction called the Diels-Alder conjugation. And the cyclopentadiene, this alkene, will react with a myelamide group in the absence of any modification or any accelerators to form this adduct here. The adduct is very stable, and the conjugation chemistry is very precise and clean. Okay, so overall, the technology gives us two key assets. We have a conjugation chemistry that lets us make very stable conjugates that are irreversible. We have an expression system that lets us control precisely where the non-natural amino acids are placed. And when we combine those two together, we have a protein engineering tool to assess what the impact of site is on an ADC. Okay, so the first thing that we looked at is positions on the Fc domain that are conserved across antibodies to see if we could find sites that could hold payloads optimally. And we chose to really focus in on hydrophobicity, the overall hydrophobicity of the ADC conjugate. So we selected a number of different positions and we generated ADCs at those positions, at those sites with hydrophobic payloads, and then used two hydrophobicity assays to evaluate the global hydrophobicity of that ADC. You can see that in red, this is a site that's well known in the industry. It's a site that's developed by Seattle Genetics. It's shown here in red as well. And this is used very extensively for engineered cysteine conjugations. But we've found sites that have, the size of the bar indicates the amount of hydrophobicity. So the lower the bar, the less hydrophobic. So we have found some sites that show much reduced hydrophobicity, and we've characterized those because we feel that those allow us to make ADCs that look like antibodies. So to test this, the first thing we did is we went to the clinic. We bought an antibody that's currently available on the market. It's an antibody that has 4 MMAE groups, classically conjugated or conventionally conjugated to the antibody. And then we generated the same versions, but with our non-natural amino acid. So the benchmark is DAR4. So we generated a DAR4 version with non-natural amino acids at 2 optimal sites. And then we generated one that had only 2 non-natural amino acids, so a DAR2 ADC. And we looked at their function and their performance. So in the top row here, you can see, oh, my mouse is not working. Okay, is the DAR2 and the DAR4 with the non-natural. This is our benchmark analyzed by HIC chromatography. And this separates molecules based on their hydrophobicity. You can see that our benchmark has multiple peaks. Conventional conjugates make heterogeneous products, so we expect to see these multiple peaks. And they represent DARs of 0, 2, 4, 6, and 8. Whereas our non-natural chemistries generate much more homogeneous ADCs. When we look at the pharmacokinetics, as I stated before, the benchmark is more hydrophobic, so it suffers in PK. It clears much more rapidly. And you can see that there's 2 lines. One represents total antibody, and one represents ADC. And the separation of the 2 lines tells us that there's loss of warhead as the antibodies circulate. However, our non-natural amino acid-based ADCs have much better PK, mAb-like PK, and the 2 lines do not separate. So we know that we have very, very stable ADCs. So the next thing we did is we did tumor growth inhibition assay. For these assays, mice are injected with tumor cells. We let the tumors grow to about 100 millimeters in size, in diameter, and then we treat them once with our drug. And we did 3 different doses testing these 3 different antibodies. The efficacious dose for the benchmark antibody is 1 mg per kg. So you can see here that at 1 mg per kg, we see no growth in the tumors. So we've cured the mouse of the tumors. However, at half that dose, you start to see regrowth of tumors after a few days. That was not the case for our non-natural amino acid ADCs. The increased stability allows us to deliver more efficiently to tumors. So we see that the performance is retained even at half the dose of the efficacious levels of the benchmark. So at 0.6 mg per kg, we still see total tumor control. The DAR4 is even pretty efficacious at 0.3 mg per kg. We had 1 mouse regrow its tumors. At DAR2, we don't see that much control. We do see some retardation in the growth of tumors, but they all regrew in the end. But keep in mind that at this dose, this ADC has half the amount of payload as this. So we anticipated that it would be much safer to use. Okay, so with this really nice technology available and the performance of this ADCs, we also wanted to tackle something that is becoming a bigger problem in the treatment of cancer. And that is that patients, once they're treated and tumors regrow, they become resistant to treatment, to the same type of treatment. And it's a growing problem in the industry. And there's many different mechanisms that they're listed at the top for why cells or cancer cells build a resistance to treatment. However, so the question is, what is being done in the industry currently? And that is, we are treating tumors with combination therapies. So I have one really good example here. This is a treatment from, a plant from Seattle Genetics that developed an ADC called Adcetris. So this is a CD30 targeted ADC that is actually on its own, it's very efficacious and performs really, really well. However, the presence of this tumor resistance is really driving the use of Adcetris with a combination of small molecule drugs, so chemotherapies. And they use 5 drugs in the CHOP family to really increase the efficacy of the treatment. Fortunately for us, our non-natural chemistry allows us to combine the non-natural conjugation method with cysteine-based conjugations to generate ADCs that have 2 warheads attached to the antibodies. So these dual warhead ADCs, and we combine drugs with different mechanisms of action to try to halt some of the resistance that we're observing. And you can see here, this is again a tumor mouse model that has a tumor that is resistant to MMAE treatment. So our DAR4 ADC is not very efficacious. However, the treatment with a dual payload ADC does lead to some tumor regressions. This dual payload ADC was also tested in a safety study in a single dose rat tox. And you can see that our benchmark antibody with DAR4 is tolerated at about 20 mg per kg. However, the dual payload has a less potent warhead in the 8th class. So we see much better overall tolerability. All right, so this is my last slide. Hopefully, I've given you a flavor for how we're using protein engineering to expand the therapeutic options for patients. There's continuous advancements with new technologies, and we'll hear about AgiCap in a second. That's also a very exciting technology. So using new technologies to generate more homogeneous and more stable ADCs that will continue to improve our ADC portfolio. Really, the real potential for treating cancer is combining our ADCs with other forms of treatment to attack tumors from multiple different angles. And ultimately combining different types of therapies to really get control and cure tumors in the end. So with that, I will leave you with my acknowledgements. I have a wonderful team of people that work with me, and I'd like to thank them.